Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to today's webinar titled Beyond Accelerated Approvals External Control Arms. In this webinar, four experts will discuss the strategic use of external control arms and, if done correctly, how they can help bring products to market in a shorter time and for lower costs. I'm Sydney Otamanchuk from the events team at scientist.com, and I'm happy to be your host for today. And I'd like to thank the sponsor of today's event, Cardinal Health, for making it all possible. I also want to take this moment to introduce our speakers from the Real World Evidence and Insights team at Cardinal Health. Our moderator for today is Dr. Bruce Feinberg, Chief Medical Officer, who has worked on the development of clinical pathways that aim to control costs, improve quality, and increase predictability. Our next speaker is Dr. Sarah Lucht, a scientist who supports studies on treatment patterns, safety outcomes, and clinical outcomes in real-world settings. Our third speaker is Harlan Hayes, Director of Research Analytics, leading a team of senior statisticians and data scientists in the conduct of statistical analysis and database management of real-world data. The fourth and final speaker with us today is Dr. Andy Klink, who is Senior Director of Business Development with over 15 years of experience in academia, industry, with health economics and outcomes research, multi-level mixed effects analysis, longitudinal analysis, comparative effectiveness, and patient-oriented research. And now, without any further delay, I'm pleased to welcome our moderator to join me. Hi, take it away.
Hey, Sydney, thank you very much. Welcome, everyone. We hope this is going to be an engaging, maybe even informative and entertaining next hour. So the agenda we'll be going through is something along these lines. We're going to be reviewing recent FDA guidelines on real-world evidence, and Sarah will be doing that. We're going to set straight the three types of control arms, well, at least three types of control arms, and discuss future trends in each. So this would be historical controls, concurrent controls, external controls, synthetic controls. I've already named four. We'll cover at least three, if not more. We'll be sharing our journey for a solution to real-world data and the challenges in them being the source of that external control arm. And Andy's going to be covering that. We'll be doing a formal Q&A session at the end, but we hope that we may be incorporating some of the questions as we go through the program. So that's what's ahead of us for this hour. Now, my job is kind of to set the stage. And in setting the stage, I want to start with some recognition of where we've been. Because really, the modern era of medicine, in some ways, begins with the randomized control trial. Beginning in the mid-1980s, and specifically in the world of cancer, we were moving from historical controls and anecdotal-based information to a process whereby we would get increasing amount of rigor in the way in which we would build our evidence and confirm and validate the effectiveness and the safety of new therapeutics, new devices, new tests. And so if we look at the left-hand panel, we get an idea of the rather extreme amount of time and amount of cost and the limited amount of efficiency that has historically been required as we moved into the randomized, double-blinded, eventually into blind independent central reviewed clinical trial for cancer medicine. And it had been for a period of decades somewhat generally accepted that it's $1 billion of cost and 12 years to move particularly novel therapeutics from the laboratory bench to the bedside. Now, this was happening in an era when we were moving into precision medicine, and that greater understanding of the human genome and the genetics and molecular underpinnings of malignancy was taking what were very large general patient cohorts, patients with advanced cancer, patients with advanced lung cancer. Then we were starting to see patients were being then small cell and large cell advanced lung cancer. Of that large cell advanced lung cancer, adeno versus squame. Of those adeno, we then realized there were at least a dozen, if not more, molecular targets that were underpinning the behavior of those cells. And we went from a disease that was lung cancer to a disease that at least has 30 types of lung cancer, or 30 types of cancers which are being cells found in the lung that are mutating and resulting in a malignant pathophysiology. What that did is increase the number of trials and make it extremely difficult both to find the patients, but also put a tremendous number of trials competing. And so over the past 20 years, as you can see in the graphic representation on the right, we've seen about the increase from about 1,500 new cancer trials annually opening to now over 6,000. So we have a process that's too costly, that takes too long, that no longer can meet the needs based on this movement into precision medicine. But it gets even more complicated and more problematic, as we see here. Those patients who are participating within the clinical trials are not necessarily representative of the patients who are being seen every day in the clinics being diagnosed with these diseases. Peter Yu, past president of ASCO, at the 2016 ASCO meeting, introduced the severity of the problem. And he said at that time that only 3% of adults with cancer were participating in trials. And of those who were, they were younger, they were healthier, they were much less diverse. They were the three he mentioned, but beyond that, they were much more socioeconomically advantaged, much more health literate than the patient population being managed with these diseases in the real world, as would be suggested by the SEER database. So we have, it takes too long, it costs too much, it can't meet the current demands of the innovations that's being brought through precision. And even when it's being used successfully, it's not necessarily representative of the population of patients, and raises that question: is the information that we're gaining really as helpful as it needs to be for the everyday patient who's being seen and treated with these diseases? Now, this prevalence of the problem goes beyond cancer, but it is most extreme in cancer because of cancer's complexity and its scope and scale, the fact that we're dealing with, in many cases, a lethal disease, and the fact that precision medicine is defining it as a much more complex entity than maybe it had once been thought. So with this in mind, it's not as if the public health, the research community, the FDA, the CDC have been sitting on their hands. And there have been a tremendous number of processes implemented with the hopes of being able to expedite the trial process in order to shorten that time frame and in order to achieve some of those goals. And so many of them are listed here on the left: fast track designation, breakthrough therapy designation, rolling review as a change in the review process, priority review. And then the last one, the one that maybe has gotten the most attention, has been accelerated approval. What accelerated approval required is that if the ultimate endpoint of effectiveness is survival, and survival can be such a long-term endpoint in many disease states and even in cancer, or had become so in cancer, did we need a surrogate marker? And surrogate markers have been extremely beneficial and helpful, but they did require post-market research, PMR, in order to further validate. But it did help to bring many innovations to the marketplace, to the patient's bedside more quickly. But surrogate markers don't work in every situation, as maybe we classically learned with Aduhelm in Alzheimer's disease in the last few years. But it can't be the only solution, because the underpinning trial design still has these issues with representativeness and cost. And even if we're trying to shorten the time frame from the classic phase 1, 2, and 3 trial design, and being able to use some sort of expanded phase 2 in order to achieve that using markers, and then continue that work in the post-market research. One way that we could start to think about what more could be done is get back to focus on the 97% of patients who currently don't participate in clinical trials. And that's where real world evidence comes in. Can we harness that 97% patient data in a meaningful way? And particularly, can we harness it to provide that control? And various methods have been looked at to try to refine that work using matching, propensity scoring, and other techniques that we'll be talking more about. And so this little bit of an introduction was hopefully intended to give you a sense of the significance of the problem, the criticality of new solutions, and start to tease the notion, could real world data provide the evidence that's necessary to start to become a control arm concept that could move us forward beyond just these processes, which are on this slide. And we're going to address one of the critical issues. And although fit for purpose is only one of the 6 choices in the poll, it's a broad topic that does address multiple issues as it relates to the way in which we think about real world data. So, Sarah, let's further inform here.
Thank you for that introduction, Bruce. As mentioned, I'll just be talking, in a broad, overarching way about what the FDA is telling us about what they mean by fit for purpose. So this is a phrase that has been used to sort of describe what they would like to see in terms of real world evidence and programs. And so just to start off about when we think about the use of RWE in regulatory decision making. So most people are perhaps familiar with the first case, and Bruce alluded to it a little bit. So after a drug has been approved, it is commonplace, typical that we would have some sort of post-market safety outcome analysis looking at safety in this broader population now that a drug has been brought to market. Where real-world evidence has been moving into is really sort of 2, 3, and hopefully eventually into 4. So 2 and 3 sort of show these situations where there's some sort of initial approval, whether it's per se an initial or accelerated approval followed by a full approval, or an approval for one indication, as in scenario 3 here, that's followed by approval in a secondary indication. RW has been used to try to support these sort of proposals or these submissions to show effectiveness here in the real world using some real world data. So where we would like to see more of it is also in those scenarios, but also in number 4. So trying to think about real world evidence as really supporting drug approvals in the primary approval setting. And I think that's where all of these things that come up in the poll, all of these potential deficiencies, really we have to think about how we're addressing them. And so the FDA has been releasing guidelines on what they view as the way to approach using real world evidence and a real world evidence program in regulatory submissions. And so the first part I'll touch on in more detail in my next slide, but really when they talk about fit for use, they're asking, are the real world data being used, are they suitable to address that specific regulatory question of interest for whatever the submission is? And in addition to that, if you do have good data and you're able to have all of the variables you need and it's of high quality, is the study adequately designed? So our data is very important, but also obviously how we design our study, how we collect it, how we analyze it. Is that all done appropriately in a well thought out way with rigor, kind of as Bruce mentioned, similar to a clinical trial? And that all of that is done in a way that can actually answer the regulatory question of interest. And then the third point here is, was the study appropriately executed? So, and again, in a similar way, trying to apply more clinical trial frameworks about study monitoring, data collection, adequate documentation of any changes that may have happened in the data or in the protocol, so that the FDA could go back and be able to see, okay, and have high confidence that this is where this data came from. This hasn't been manipulated. It hasn't been sort of cherry picked, and really being able to adequately document real world data in a way that's more rigorous. And so the main point from the FDA that I'll touch on today is this fit for use. And so when we think about fit for use, the FDA has really used 2 buckets to think about that. So the first being here on the left part of the slide is reliability. And I guess I operationalize this more as data quality. So thinking somewhat about data missingness. So data accrual is the data there. If you have a data set or an EMR or a claims data set where it has missing data on a certain outcome for 50%, that's perhaps not the most promising sign. But so is the data there, but also is it of high quality? Do you trust it? So if it's in a claims database and it has a medical code, does that medical code adequately represent the underlying concept? If you have a patient reported outcome, how well do you trust that? Where's data collected in a standardized fashion? Where is the data coming from? All of these questions fall under then reliability. So what level of quality, how trustworthy sort of is that data? And the other thing to think about in terms of data being fit for use is relevance. So on a very basic level, is the data that you need captured and available in that real world data set? And that might be different it's obviously different for different questions, but will be different depending on your data source. So claims data sources will have different data captured perhaps than EMR data sources, which then might have different data collected in its structured versus unstructured areas. So you have to think really critically about your data source when thinking about relevance and about your research question. So we know from randomized clinical trials that they're inherently randomized. So that exposure is randomized. And the main hope for that, the reason for doing it is so that you can see an unbiased estimate of that association between your exposure and your outcome. Real world evidence is obviously does not come from a randomized population. These patients are likely getting in oncology that first line drug for a reason, that maybe they have certain comorbidities that mean they can't get that other drug, that they have certain insurance or certain financial concerns or a different medical history. And so we know that. So it's not just about being able to capture perhaps the treatment, but also thinking about potential confounders. And so that in real world evidence is really important when you're thinking about the data source, that if you do say have a comorbidity that you know is influential in terms of that treatment selection or your exposure, but also influential on your outcome, whether it's associated with mortality or progression or something like that, then you do also need to be looking for that data in your real world data set and to make sure that you have coverage of all of the relevant data so that later you can adjust appropriately for potential confounding or other biases. So practically, when you think about this, if you thought, oh, I would really like to use real world data or real world evidence to support a regulatory submission, what does the FDA sort of tell you to do? The answer is to do things in a rigorous fashion. So to approach it, not sort of in a, we have some clinical trial data, now we'd like to add real world evidence, but really to start with including real world evidence from the beginning and sort of thinking very strategically through all essential elements of the study here. In this first bullet, sort of predefine what sort of design would be appropriate here, what data source are we thinking of using, how are we going to analyze it, how are we going to address those potential biases, selection bias, data representativeness, missingness, potential confounding, and how are we going to conduct the study? So in developing this protocol to use real world evidence, really also being very prescriptive in terms of describing. So how are the elements going to be ascertained from the data sources? How are you going to say that that outcome is truly representative of what was maybe captured in a clinical trial? So thinking through each aspect of what you're going to get from that data source and trying in this third bullet point really to address the FDA's concerns. The concerns that were raised in the poll. So is that data reliable? How do you know that? And do you have all of the relevant data? So really trying to put yourself in the position where you can make the strongest case possible that the results you're seeing using your real world evidence are indeed good. That you don't have to worry that much about missingness because of X and you've adjusted for that, or you've done imputation and you're sure that your pattern of missingness is appropriately assigned, or that you've adjusted for potential confounders, that you're going to run the appropriate sensitivity analyses. And all of these really are recommended to be done before you see any sort of data or results. So that you lose that aspect, I think, or the concern that with real world data, it's accessible, it's available, of cherry picking, that we reran the model until we got the right result. And that's really what the FDA doesn't recommend here. That really we are approaching real world evidence with the same sort of rigor and strategy that we would approach a clinical trial, and thinking very thoughtfully about it. And then this last point here about consulting is about thinking about conferring with the FDA prior to the start of the study. This is highly recommended, but I would say it doesn't necessarily need to come here after you've done the first 3, but could be done early and often in a way that gets that important feedback and allows you to address any gaps ahead of time, again, before you've really done any analyses, seen any results, interpreted them, so that you really are placing yourself in the best position possible to have the strongest real world evidence to support your regulatory submission. And so I think, Bruce, that brings me to the end of my slides.
So Sarah, that was great. And one of the things that immediately comes to mind as I digest what you presented is this recognition that among the many things that are changing in this very dynamic environment of clinical research, and particularly cancer research, is that we are in an era now where we're starting to introduce curative intent treatments. And there was some effort to do that in adjuvant therapies, but even in the advanced setting. And we're starting to see that happen with immunologic-based therapies in melanoma, but with the advent of genetic intervention, cellular therapies, CAR T to be specific, we now have late introduction of treatments that are potentially curative. And when you deal with curative treatment, the notion of the delay of the potential for 12 years for a phase 3 and the ethics of having to perform that type of research, where we had palliative, for the most part, palliative efforts in our therapeutics. And now we have an era where we're entering into curative therapeutics. And it seems to me that there almost is no choice. Like, there's got to be another way. Now, so it seems like the real world data is the right way to go, if we can figure out how to do it in a compelling fashion.
Yeah, I mean, I agree. I think, because I didn't really touch on sort of the scenarios in which you would be considering real world data, but I think rare disease, again, if there are no other treatments, sort of what would you randomize a patient to? So you need a comparison group. And in the same way with curative treatments, you can ethically, if there's no other treatment, you can ethically, for rare disease, say we'll give you nothing, because something's probably better than nothing, you would hope. And in the same way with curative disease, we're like, oh, we could cure you, or you might get placebo or some other standard of care that we know is not going to work. I think those are the real spots too, where people are really interested in how can we use this real world data? How do we develop the rigor and the methods and the submission, the evaluation, all of it to make it the most sort of convincing and scientifically sound way, particularly when there are no real good alternatives to be offered by clinical trials or randomized clinical trials in their current sort of form. Mm-hmm.
Thanks. All right, so we're gonna move on, and we're gonna bring Harlan in. And Harlan, this, you would need an hour plus to even touch on this. The simple part of what you're going to do is hopefully explain to us all these different types of terminology, and do they really represent something different? Or are they all referring to the same thing? But the synthetic control, the external control, the concurrent, the historical, what they all mean. And then beyond that, hopefully get into a little bit of the underlying data requirements, and what will that allow us to do. So you don't have a lot of time. Get right at it, and let's see what we've got.
Thank you, Dr. Feinberg, for the introduction, and to Dr. Lucht for kind of setting up both the need for this discussion as well as some of the ways in which we're thinking about this. So I want to start with definitions, as it turns out that the biggest thing in the control arms is how do we even talk about this? And what are we hearing from regulatory agencies like the FDA and their guidance towards industry and other components? So I'm not going to read these exactly, but I want to talk through a little bit of each of this. So the historical control is really this idea of in the past. So things that have happened and occurred. And as we all know right now, a lot has happened in the past to change treatment, to change access to care, to change what we were doing in research and other components. And so the historical control, while good in a real world evidence area, is limited by how similar was yesterday to today. And this really does result in a way for us to really have the ability to do comparison groups as we're starting to look. And it's great for sort of the exploratory. It's great for understanding unmet treatment need and other components, but that's not enough for regulatory submission. We need something that still has a little bit more scientific vigor to it. And so we look at these other two that oftentimes actually get said as the same thing, but there really is a subtle but very distinct difference, which is this concept between an external control and a synthetic control. Frequently, you'll hear people talk about them in the same way, but the big difference is in what groups are eligible to be in each of these. So in an external control, we want to find individuals who are outside of the trial. They're outside of this, and they're receiving tests and protocols that are similar to what actually happens within the control arm, within the case arm. And so as we start to look at this, we can say, well, what are some of these things that occur? Now, sometimes, this is where it gets confusing, an external control could be a historic. It can be a historical control. Or the more ideal is to have a concurrent external control arm. What does that mean? I mean, it's find individuals that at the same time when this trial was occurring were receiving therapies. And what we're doing in that is to try and find them in another setting, an additional place. It gives us a viewpoint into what were things happening at the same time. It takes out a little bit of the bias of that temporal component of what's happened in the past, but we have to make sure that the data is there. And we'll talk about that here in a minute. The last one I wanted to hit is this idea of a synthetic control, right? Sometimes it's synonymous with external control, but I really actually like the FDA guidance that separates them. The extra layer that it adds in is it is similar to that external control, but that the data come from clinical trials only. So this means reusing or starting to look at controls that I've collected for study A and start to use them in study B. So this is sort of a little bit of that idea that we start to harness what's already been collected. So one of the things that Dr. Feinberg hit so pointedly is time, expense, and difficulty locating patients. We all know that it's difficult to get individuals consented through the protocol. There's a washout period, getting them to finish everything. That limits our population. So if you've spent that time and money to gather individuals who have received either the standard of care, which is traditional in oncology, or no care, which we really don't see very often anymore, you could use those individuals and reuse them into it. So you have clinical trial A through X. You're able to identify those external patients because, again, they aren't part of your trial. They were in a different setting. And you build out now through a sampling and weighting methodology, a simulated patient pool as if these were now your controls. This is a place where, in some aspects, this is real world, right? But it's also been very heavily controlled because it's still coming from that clinical trial mentality. So what if we went to a pure real world version of this? How does that change things? So I like to think of the external control, whether it be concurrent or otherwise, as borrowing from existing data and matching to the trial criteria. This data is being generated for a variety of uses, most importantly, care that's being utilized in care delivery. So there's a lot of information that's there. However, and this is a big however, it's highly dependent on the sources that you're pulling this from. Claims are fantastic for understanding where things have occurred and a little bit of what has occurred, but the details just aren't there. An EMR in the structured fields will give you a little bit more of the details of the actual lab values and other components that will feel more like a clinical trial, but you had no control over how often those labs were captured. So maybe your clinical trial protocol looks at it every 4 weeks, and in the population, you're seeing it every 6 months. And so it dramatically impacts your ability to kind of adjust for these individuals. Now, once we start to add in EMR unstructured, so these are the notes, the clinical aspects, the thoughts and patterns that are occurring within an EMR that are captured in an unstructured format that requires some sort of natural language processing, or in the newer world, large language models with generative AI, to identify key components in there, we may start to identify even more data that's readily available. Also, you don't get the patient voice in those. So adding in that PRO component to it that may already exist or may not exist could help to augment that. And holistically, all of those together start to form a much more robust external data pool that has all of the critical data elements and allows you to synthesize and replicate and create similarity to the original criteria of who was in there. Now, we don't want to just limit it to those. One of the advantages that we find in external data like this is our ability to oversample individuals within. So we're able to identify individuals maybe from minority populations, which we identified earlier on, tend to be missing. And the advantage is you can utilize those individuals to do the whole study and then do subgroup analyses to actually look and say, well, what was actually occurring amongst this minority group, while still having the ability to do a matching in your control and treatment arm at the analytic level rather than at the study design level. So it gives you the ability to do that sensitivity analysis that allows you to really highlight what actually occurred in this population that is super similar to my treatment arm and identify those individuals. Here you need lots of data. You need lots of information to be able to go do this because you're going to be throwing out a lot of people that just don't make that cut due to those data dependencies we talked about earlier, both from a quality as well as similarity perspective. I want to leave you with one last one, though, to think about, which is what does a true synthetic control look like? So the word synthetic to me is a little bit tricky because it's something that we use right now, but true synthetic data, as kind of developed by the Royal Society, says data that's been generated using a purpose-built, that's really important, mathematical model or algorithm with the aiming of solving a data science problem. And I would say a research problem in this. So the future of this is to add in some of these large language models, some of this generative AI. So let me describe to you a world where I think that we're going to move towards in the research area, which is this concept of a digital twin. So we can take Harlan's record and de-identify it so it's no longer Harlan and create a digital twin and then allow for simulation to experience that growth. So they take my real record, de-identify it, and now I have a twin. But I allow that twin to mature through data and application to create future state data elements, outcomes, and other component through replication. It will predict the behavior of the choices I've made in the past towards choices into the future and what may occur to me. So I no longer need Harlan's data, and it's no longer, or is it, Harlan? And that's one of the ethical things we get into is that data, in fact, still the same person? How much quality does it have? If we're utilizing prediction, how accurate is that towards future things like outcomes? And we've yet to really receive that guidance from our regulatory bodies, but I think they look to the industry to identify what is potentially there. So that's where we get into this idea of the bias and the quality that goes into it, because we have to understand how well does this really represent the ideal future state. In order to do that, and a lot of things, I highly recommend what we would call a human in the loop aspect. So allowing the data to mature and change through these digital twins and other components through various synthetic data generation, but having a human in the loop to say, does this clinically make sense? Does this accurately represent the current state of treatment? And by sampling some real patient charts and adding them into our digital twin charts, we start to say, are these significantly different? If the answer is yes, we've done a poor job of our replications and making synthetic data. If the answer is no, what we've done is increased our power and our ability to start to look for some of these more unique things. And I'll leave you with this idea that as we move into this AI-driven synthetic controls, that we really need the global community to start to look at what the privacy concerns are. How will regulatory agencies adopt this? Will I require a still traditional role of a control and then add in some synthetic data on top of that? And how will that be part of my submission? How will we address privacy? Because as I created that digital twin, it was in part Harlan. But at some point in time, it starts to move away from being there by potentially we can add in noise. But if we add in noise from a privacy concern, guess what that happens to the research? The noise is also added in and we lose signal. And the last part is this idea or concept of the chain of custody. So how do we ensure true privacy and understand who has access to this? And what is the level of risk we're willing to tolerate as we start to build out these different areas? So hopefully I've left you now with a little bit of the various ways in which we start to think about these different clinical trials and the different control arms from a real world setting that we're able to identify.
So Harlan, that was a mindful. And so the first thing I want to say is that, and let the audience know, that you are not a frustrated science fiction writer. But the things that you are talking about are actually things that are actively being looked at by the FDA.
That is correct. And especially as we start to look at these large language models and understanding how to do this. But historically, right now, today, submission of these external control arms is a thing we can do. In fact, it's a thing we should do. And the FDA is looking to us to help reduce the burden of disease and speed up those clinical trials by using real world evidence right now.
All right, and as a direct segue to that comment, if you could just give us a minute, which is not fair, but on this idea of matching, using more, even just in the basis of like propensity scoring, but with some broader approach to that concept of matching that underpins how we get that external control to be closely representative of the patients in the trial.
Yeah, I think ultimately what we're trying to replicate in that is the idea of selection and randomization. And the way in which we typically do randomization is we find two individuals who are matched at explicit variables, be it sex at birth, age, and maybe say stage for a particular cancer. And by doing that, we're eliminating some of the bias that goes into the decision, and we randomize them to control or treatment. In the real world setting, what we need to do is we need to create as much similarity between the chart, the EMR, the claims, and those individuals who are now digital versions to as a real person would be in the clinical trial, and remove as much of that bias up front by, again, propensity score matching. So that's a way of statistically and analytically, by doing hard matching like we do in traditional trials based off of various key characteristics, to ultimately remove as much of that bias into the decision making, so that afterwards we're really looking at just the effect of the treatment, not the effect of the treatment decision, but the effect of what was actually part of the protocol.
Thanks, Harlan. All right, Andy, you get to close out before we go to Q&A. And you're going to, I don't want to say it's not fair, but Harlan, but you're going to bring us back to reality, and kind of what's being done today, kind of what you're doing as a researcher that's allowing you to use real world data to be translated into quality evidence that's fit for use for FDA submission. Take it away.
Thanks, Bruce. So yeah, we've heard from the kind of the FDA perspective, some definitions to lay the groundwork here, but what do we need to actually see in order to create an external control arm? What does good look like? Which data sets meet these minimum requirements related to these 4 areas that we consider when creating an external control arm? There are obviously more than 4 areas here, but I will distill a lot of what my colleagues, Sarah and Harlan, have introduced into these 4 buckets here. So starting with data completeness, in an ideal setting, we'd have a data set with no missing data. For each patient in that data set, we would have non-missing data for every variable that's included in that analysis, from baseline characteristics to covariates to consider potential confounding to the outcomes of interest. And as what was just mentioned, in external control arms, because they're typically matched to their trial arm, we need non-missing on those key matching variables in particular. Next up is endpoints. So now that we have a data set that we've identified with acceptable non-missingness, we need to consider how the endpoints have been measured in that data set. First, do the endpoints exist at all in that data set? The example I often think of is data progression documented or available in that data set? Or do we need to rely on a surrogate or a proxy like time to discontinuation of therapy? We also need to consider how those endpoints in that real data set are measured, and how do they compare to how they were measured in the trial? Were they measured in a similar way? And if not, how might this introduce bias, and how could we methodologically account for that? Representativeness. Does the data set contain patients that represent the target population in terms of age, sex, region? How about prognosis, stage, or number of prior therapies? Further, does this data set allow for assessment of relevant underrepresented patient populations? So even if we were to emulate an age distribution that looks like the trial population, do we have enough patients over 70 years old, for example, to assess characteristics and outcomes in that important subgroup? And of course, cost and time is a big consideration. External control arms represent substantial cost and time savings, where timelines, when leveraging existing real-world data to create an external control arm, could be measured in months. Trials, RCTs, could be measured in years. Similarly, for cost could be tenfold in an RCT compared to leveraging real-world data to generate an external control arm. Ultimately, I think external control arms represent a pathway to get key therapies sooner to the right patients compared to traditional randomized control trials. So next, I'm going to dive into each of those sections here, starting with representativeness. We took a look at several real-world evidence studies that we've conducted over the years and aligned them with their respective pivotal randomized control trial, as well as SEER data, which represents the general US population. We expected differences, and here we see differences in representation in patient age, ethnicity, sex, and race when we look at real evidence compared to their RCTs. In each case here, we have greater representation and distribution in the real evidence population compared to RCTs. So why does this matter? First, we want to be able to create an appropriate comparator arm in our external control arm that can be appropriately compared to the trial arm, so with or without additional matching or trimming of that external control arm. Second, ECA's external control arms may allow for, again, that assessment of important underrepresented subgroups. So historically, older patients, patients of poor health status, patients of certain races may not have been adequately enrolled in and represented in their clinical trials. In the external control arm setting, we often find those patient populations represented, allowing us to do additional assessments of both patient characteristics and outcomes in those important subgroups. Here I'm going to focus on endpoints, and perhaps the most important aspect here is how endpoints are measured. The same topic and framework would also apply to the exposure variable, how diagnoses are determined in identifying your patient population for your external control arm. But here I wanted to focus on how endpoints are measured in the real world setting compared to how they're measured in RCTs. So take, for example, response to therapy shown on this slide here. In an RCT, PET CT scans may be sent to blinded independent central review for review by 2 radiologists who apply strict RECIST criteria in the case of solid tumors here, or Lugano criteria in the case of hematologic malignancies in order to classify response to therapy. This may be the primary endpoint in a clinical trial. So we need to get it right in the real-world data setting. In RWD, we may be limited to what was charted by the physician as their response to therapy, and not by more strict criteria that the trial used. We've developed real-world measures of responsive therapy that mimics how they're measured in a trial. RECIST criteria involve data elements related to tumor location and size. And if we measure the change in the sum of diameters over time, we're able to apply RECIST criteria similarly to how it's done in a clinical trial in order to classify responsive therapy in a similar manner. These data points exist in patient charts outside the trial setting, despite them being used to assess response in routine clinical practice. Through this work and similar work in the hematologic malignancy space, we've demonstrated that the application of RESIST criteria or Lugano criteria to data elements available in patient charts has produced measures of response that are more closely aligned with blinded independent central review compared to physician charge response. So understanding these differences in how these endpoints are measured will help regulatory agencies assess potential biases from real-world data when creating external control arms. I'm going to spend the next couple slides here really focusing on data completeness. I wanted to do so because this is really where I wanted to highlight a method that we've developed over the years that has greatly reduced the amount of data missingness in real-world data sets that we have used to generate real evidence. So one traditional challenge in leveraging existing real-world data sets is the reliance on data appearing in structured fields. If it's not in an unstructured field, some method of curation is going to be needed. One approach that we've taken is a physician-led chart abstraction. So here, the treating physician navigates their own patient's medical records to abstract data elements, regardless of the EMR platform, regardless of whether it's in a structured or unstructured field, or whether it's even within their primary EMR data system. For example, physicians may have access to PAC system to retrieve PET CT scans or pathology reports from outside sources. Even beyond what they can get from a patient chart, we can also assess physician assessments in terms of reasons for discontinuation or reasons for dose modification, which can readily be captured from the treating physician at the time of data abstraction, creating a really robust data set suitable for an external control arm. Here I'm going to use CAR T therapy as an example here because it really presents a unique patient journey that involves multiple sites of care. A patient may start out in a community oncology setting in which their treating physician is assessing them for the fitness and eligibility to potentially undergo CAR T therapy. Once that referral is made, their care may be transferred to an academic medical center. They may ultimately receive CAR T therapy in an inpatient setting, and then follow-up care and patient monitoring taking place back at the community oncology setting. This really illustrates the potential, the likelihood of gaps in the data and the potential for missing data. So how do we account for that? This is an example of work that we've completed that really has demonstrated how we were able to piece those data points across multiple sites of care. This was using a physician-led chart review where the treating physician, or the referring physician in this case, had access to the key data elements that we needed in order to construct that patient journey and reduce data missingness. Here we see for assessment of various adverse events, 91% of patients had anxiety assessed, 95% of patients had cytokine release syndrome assessed, and reported that 79% of patients had documented CRS. On the right-hand side, we see 100% capture of the actual CAR T product that was used for therapy. So again, this community referring physician has access to this detailed information, including dates of infusion. So 100% capture on when that CAR T infusion was performed. We also saw a really high capture of supporting therapies and procedures like leukapheresis in this treatment journey. So through this physician-led chart review method, we've addressed data completeness, representativeness, endpoint assessments, and of course, time and cost to create an external control arm suitable for regulatory decision-making.
All right, Andy. That was a great summary of not just your work, but bringing all the pieces together from the prior presenters. And I think what we'll do is, given the time, we'll move into Q&A. For those who do have a question, there's still a chance to get it, well, depending on how fast you type, to type it in now, and we'll get it into the queue. And for those who have already put a question in, sit tight. We should be hopefully getting to most of them. I'm going to kick us off, and let's bring up all the presenters on screen so we can all be seen. And I'm going to start out with a question, and Sarah, I'll give you the shot at this. But you mentioned one of our common responses to clients about when do you consult the FDA, and the answer is early and often. And so the question from one of our participants is, how early is early? Is early like pre-biologic license application early? Like when is early? Is there anything that is there any concept that's too early?
I guess, right. I mean, you touched on it in your last point. I haven't seen, which again, there have been a lot of guidelines being released by the FDA related to RWA in the last couple of years. So I haven't seen necessarily that there's a prescriptive, like, this is exactly the right time in the process. But I think that no time is too early, especially if you have a situation that you think is amenable to an external control arm using RWE, kind of like we mentioned, maybe rare disease or an ethical sort of concern related to some other aspect, or if you have just a situation where you have pretty concrete outcomes that are captured well in real world data. I think asking as early as possible can only set you up for more success. Maybe from the beginning they say, I'm not sure, and you revisit it at a later phase if you think, things have advanced. But right, I guess I personally wouldn't say that any time is really too early to at least get some initial feedback, to say, we've been thinking about this. This is our idea. We thought this would be an interesting situation to use. Because then you do give yourself, if they say, maybe not right now, you still have time if it continues on, to a further phase, a further part of the process to maybe revisit it if you feel like evidence has increased or the methods have improved, or that you have better evidence on your hand that it is suitable. But I guess I don't see any time as being too early to start sourcing that information.
Yeah. So
one thing I was just going to just kind of in a broader sense of applying real data or real evidence, I think more broadly could certainly be done earlier on, not just in the context of an external control arm. So when we think about patient preference or really getting the patient voice early on in really that development of the actual product or the molecule. So we really want to kind of infuse real world evidence in general really early on in the process. But when we're talking about specifically external control arms, oftentimes is, to align with the timing of when data of the trial is going to be available so that we then have that contemporary external control arm.
Mm-hmm.
Thanks, Andy. Thanks, Sarah. And I will also hearken to one of our colleagues, Scott Swain, whose past career was at the FDA with the CEDAR group. And he reminds me often that there's no penalty. The FDA doesn't penalize you because you came too early. They welcome it. They'd rather it be early than too late. And so there's no penalty box. If you go and they think you need more before you come back, they'll tell you what it is they want. All right. So Harlan, I'm going to you next. So you touched on a lot of AI. It was interesting, and it felt a little like, more than just futuristic. That's why I referenced the sci-fi. Also because I started watching Three Body Problem because I really love the book. But anyway, but when you talk about kind of the AI use, there's this notion in AI of hallucinations, artifacts and data that aren't real. And so it gets back to like, is it Star Wars or is it, are we talking reality based, and how quickly are we talking it, and how are things like that to be addressed?
Yeah, there's a number of tools out there right now in the large language models, which is the LLMs that I referenced, that start to get at this. And the hallucination is things that aren't real, but it's expecting that you're expecting an answer, so it makes up an answer. So right now, the easiest way is to create and only utilize products that have huge amount of traceability and have that human in the loop. So have a clinician like yourself or others that can go into it and say, well, this person is saying they have this particular rare stage combination of therapy and other components. Can I trace that all the way back to the real data? Can I see where that came from? And can I go read that portion? And if I can't, you should be avoiding it right now. If I can trace it, that gives us a little bit more credit. Now, I do think that we're in a place now where it's not going to be ready for FDA submission for some time, but it could be utilized for real world evidence studies to just get an idea of the types of things that are occurring that are locked in those structured notes. And you could do that. Honestly, there are platforms that are doing it today and doing it fairly well.
All right. So maybe time for one more, Andy. We'll go to you. Based on the work that we've been doing with the FDA, what do you see, or how would you describe the biggest barriers that the FDA is identifying to us for accepting external control arms as part of the evidence package?
Yes, I think we introduced, our 4 areas that we really consider when developing an external control arm. But one thing that maybe Sarah touched on a little bit when talking about really infusing scientific rigor in constructing and applying an external control arm really is that the research question that the external control arm is trying to answer aligns with the regulatory question. And from what we've heard from the FDA, members of the FDA, that's often there's often a disconnect in those 2 questions, and that's really what we want to get right early on. And obviously, meeting with the FDA early and often certainly helps align those 2 questions.
All right, thanks, guys. You were great. Really appreciate all those who tuned in.
In closing, we hope you enjoyed this HealthEconomics.com webinar sponsored by Cardinal Health, and I hope you enjoy the rest of your day.